Welcome, everyone. This is the latest edition of the JMBBM Frontiers webinar, the February edition this time around. And it's my great pleasure to introduce the moderator today, Professor Michelle Oyen. I think Michelle is one of those people where they say she doesn't need an introduction, but I'll just say a few words. Michelle is professor of biomedical engineering at East Carolina University. She's very well known for her work on tissues, mechanics of tissues, indentation, and has been, I think, active in the field for many years and has been a leader in the field for many years. And in the recent several years, she's become very interested in the mechanics of pregnancy and women's health. And that's the topic she has agreed to organize today. And so she has brought to this discussion really an amazing set of speakers on the topic of biomechanics of pregnancy broadly, and related topics. And I think this is a very timely and very interesting direction where biomechanics is heading. So the frontiers aspect of this topic is really there. And I'm very pleased to hand it over now to Michelle, and she will be moderating, keeping time. And I hope you all have fun and enjoy the presentations. Thank you, Michelle.
Thank you very much, Marcus, for the introduction. I'm really pleased that coincidentally, and I didn't realize this until yesterday, that it's the day for celebrating international women and girls in science and engineering. And so coincidentally, we have a completely female panel of participants today talking about women's health and bioengineering. So it's one of those things I couldn't have planned, or maybe I should have planned, but I didn't. So I'm going to introduce the first speaker. So we'll have two speakers. Each is going to speak for about 25 minutes, and then there'll be questions. And the first speaker is Dr. Kyoko Yoshida, and she is a senior scientist in the Department of Biomedical Engineering at the University of Virginia. I have the pleasure of having known Kyoko since she was a graduate student working with Kristen Myers at Columbia University. And I will turn it over to her. Please go ahead and share your screen and start, Kyoko.
All right. Okay, so thank you so much, Michelle and Marcus, for this really cool opportunity to reach a very wide audience today. And it's been fun to, as a graduate student, I visited Michelle's lab a long time ago and learned how to nano-indent. Unfortunately, I didn't do much nano-indentation after that, but it was still really great to have known Michelle for so long, and she's really been a great mentor as well. So today I'm really excited to share with you the work I've been doing, understanding pregnancy-induced heart growth. So today I'll jump into my talk on the multiscale model of pregnancy-induced heart growth, where we integrate hormonal signaling with mechanics. So during pregnancy, we see big changes in both the mechanics and the biology. So on the mechanics side, we see increased loading and growth of virtually all maternal soft tissues. So in today's talk, I'll be focusing on the mother's circulatory system. So the mother will undergo significant changes in her circulatory system, including a large drop in the systemic resistance. So this occurs as her aorta starts to relax, and then as the fetal circulation also starts to expand. And at the same time, we see an expansion in the total blood volume. So total blood volume increases by about 50% throughout the course of a pregnancy. I'll also be focusing on the maternal heart. So the maternal heart actually pumps more blood during pregnancy, about 45%. And so this is characterized by an increase in the cardiac output. So cardiac output is actually a measure of how much blood the heart is pumping to the body as a unit of time. And in response to these cues, the maternal heart grows in size. So it grows about 20% in mass. And this occurs both to support both mom and baby for a healthy pregnancy. And then on the biological side, we of course have these big surges in the pregnancy hormones. So in today's talk, I'll be focusing on two pregnancy hormones, estrogen and progesterone. And here I'm just showing how their levels increase throughout the course of a pregnancy. And so why is this important to study? Why is it important to study how a mother's heart and circulation adapt during pregnancy? Well, it's important to study because heart problems are the number one causes of pregnancy-related deaths. In 2015, there was a study that showed that maternal mortality is rising in the United States. And if we look at the reasons why mothers are dying, we see that we're doing well in treating certain conditions, so including hemorrhage, hypertension, and infection. So these rates are actually going down. However, the cases of cardiovascular conditions, as well as cardiomyopathy, so this is heart failure that's associated with hormone or associated with pregnancy, is actually rising each year. And so the question that I'm really interested in answering is, how do these big changes in the mechanics and in the hormones or in the biology interacting to drive this really important tissue growth and remodeling process during pregnancy? So individually, we know that if we induce tissues to high levels of stretch or stress, the tissue will respond by growing and remodeling. And we also know that hormones can also have this effect. So treating cells or tissues with hormones will actually lead it to grow. And so what does this interaction look like? But then also considering how this growth and remodeling process then feeds back to alter the mechanical states within the tissue. So for example, if a mother's heart grows in size, but is subjected to the same amount of pressure, the cells start feeling less stretch or less stressed. And so considering this feedback loop is also important if we want to understand how it's adapting during pregnancy. And so a little bit more detail about how hormones and mechanics influence heart growth. So in particular, estrogen, one of the pregnancy hormones, has been shown to have a role in preventing growth. So here I'm showing an experiment in a control mouse. So this is a mouse heart, a normal mouse heart. You can infuse mice with high levels of hormones. So this is, in this case, is angiotensin 2. So angiotensin 2 is a heart failure associated hormone. But if you infuse mice, this leads to the heart actually growing. So the heart will actually get bigger. However, if you infuse mice with angiotensin along with estrogen, what happens is that this growth is actually attenuated. So you can see that the size of this heart is more comparable to the control heart. On the other hand, the other hormone, progesterone, has been shown to have a role in inducing growth. So this has been shown in an in vitro study. So this is a study where cardiomyocytes or heart muscle cells are isolated and put in culture. And when these cells are treated with progesterone, you can see that individual cells get bigger. So if you compare a cell in the left image here and the right image here, you can see that the individual cells get bigger. And the same group also repeated the study in vivo in mice by infusing mice with high levels of progesterone, and also observed that the LV mass, or the left ventricular mass, actually grew by about 10%. So it does induce heart growth. It's also very well established that mechanical overloading the heart also leads to growth. So there are a couple different ways to overload the heart or mechanically overload the heart, but today I'll be focusing on volume overload. So this is when there is more volume in the heart than it's used to that leads to this mechanical overload that can lead to changes in heart mass or growth. And so you can see that in this particular study conducted on rats, they saw about a 2-fold increase in heart mass. But what's particularly interesting about this study was they also looked at the combination of volume overload and estrogen. And so if you induce this particular condition, you can actually attenuate this heart growth from occurring. So it's very similar to this angiotensin study here. And to me, this was really interesting because this is actually showing us experimental evidence that hormones and mechanics can actually interact to influence heart growth. And so with these interactions in mind, what I'll be talking about today is a multiscale heart growth model that I'm developing to understand heart growth during pregnancy. So in general, we have 2 computational models. So this model on the left here is a signaling network model that represents the cellular and the intracellular scales. And the general idea here is that this model is able to take into account the changes in hormones that occur during pregnancy. And so I'll talk into a little bit more detail later on, but what we're simulating here is all of the different signaling pathways that can occur within a cell that lead to changes in the cell area or the cells actually increasing in size. We then have the second model, which is a compartmental growth model. So this is a mechanical model. And the main idea here is that we're able to take into account the changes in mechanical loading that occur during pregnancy. So for example, that increase in stress blood volume or increase in blood volume, as well as a decrease in resistance, as well as growth. So we can take the cell area related to ventricular growth or tissue level growth. And what this model is able to do is to take these 2 changes into account and then solve for multiple heartbeats, so the mechanics of multiple heartbeats, to calculate the changes in stretch that occur, which are then fed back into the system, the signaling network model. So for this particular study, I built this signaling network model with 4 main inputs. So the first two inputs are progesterone and estrogen. So these are the pregnancy hormones. And we also included angiotensin II because we were simulating volume overload, and we know that angiotensin II levels actually rise during volume overload, as well as a mechanical signal stretch. The main output from this model is cell area. Again, so this is representing how much growth is occurring within the cell and within the heart. And so each of these lines that you see here are reactions. And so basically what this model ends up is a system of normalized Hill-type differential equations. So Hill-type is really giving us a sigmoidal curve that we know occur in these biological processes. And then the normalized part basically describes the fact that each of these species, so these can be receptors, for example, or kinases that are have a role in signaling within the cell. And these can take a value between 0 and 1. So 0 represents no activity of that particular species, while 1 represents maximum activation. And so you might be wondering how we decided to wire these together. So how do we decide on these reactions? So we basically decided on these reactions by adjusting them to match in vitro experiments. So in particular, I looked at experiments where cells were treated with progesterone, estrogen, or the combination of the two, as well as angiotensin and angiotensin plus estrogen. And then I should also mention here that this mechanical signal stretch here is also part of this reaction. And so this was actually based off of a previous model that was published by Rayal et al. In Jeff Sossaman's lab at the University of Virginia. And so this is actually a larger scale network model that is a little bit more comprehensive that was developed in the context of heart failure. So this compartmental model, the mechanical model, is what we call the compartmental model of the circulation in the heart. And basically what this model is able to do is we're able to simulate blood flow within the circulatory system, and then how that actually loads the heart. So you can see here that we have resistors and capacitors. So these actually represent the aortas and the veins. And then we also have valves here. So these diodes are actually valves that allow blood flow only in one direction. And so with this model, what we're able to do is to adjust the parameters to match a mechanical state. So here in the black dots, I'm showing experimental data of a pressure volume relationship for a normal rat. And then in the solid line, we're showing how our model is able to capture this data. And so here we have end diastole, the valves close in the heart in the left ventricle and starts isovolumic contracting. Then the valve opens to allow for ejection of blood into the rest of the body. Then the valves close again, allows it to relax. And then as the valve opens, it fills back up with blood again to repeat this whole process. And what's nice about this particular computational approach is that it's actually very computationally inexpensive. So sometimes people do these types of models with finite element models, but we're actually making a thin-walled assumption of the left and the right ventricle. And this is a common technique that we use in our lab in order to capture growth within the heart. And the other thing that's nice about this model is that it's actually very easy to adapt it to a particular experimental condition. So in order to actually simulate volume overload, what we did for this study was actually add a connection between the aorta and the vena cava or the veins. And this is because this is actually how it's experimentally induced. So a fistula or a connection between the aorta and vena cava is introduced in order to divert blood flow into the RV and into the LV, so the left and the right ventricles, in order to get this volume overloaded state. So in red here, I'm showing in the dots the experimental data from volume overload in rats via this aortocaval fistula. And then in the red line here, I'm showing how we can adjust this resistance parameter in order to capture this change in the mechanics. So this really allows us to quantitatively match the mechanical state in a particular condition. And so one last piece of this framework here is accounting for growth. So in order to account for growth, we use a kinematic growth framework that was introduced by Rodriguez et al. In 1994. And so the reason why we need this particular framework is because when we observe tissues in vivo, before it's grown and after it's grown, the observed shape change is actually a function of both a change in loading. So the tissues can be under two different loading conditions or pressures, as well as changes in growth. So this is individual cells or tissue constituents actually getting bigger. And so in order to decouple these two effects, what we do is we first unload the ventricle so that it's in a stress-free state. Then we apply growth in the stress-free state. And so here, this growth is a function of cell area that's calculated by the network model. And we apply this as an inelastic growth stretch. And so once we apply this, in a stress-free state, we can get incompatible growth. But then we can apply an elastic stretch in order to enforce compatibility. And so in a stress-free, so when we do this, this can actually introduce residual stresses, which is what I'm showing here. So you can see we have some compressive residual stresses within the cylinder and tensile residual stresses on the outside. And this is a product. So this is a multiplicative framework. So the total deformation to get from this state to this state is a product between this elastic stretch that enforces compatibility as well as this inelastic growth stretch. Then we can reload the ventricle or the heart to get back to this loaded grown state. And so again, this allows us to decouple these two effects that lead to changes in shape. And so now that we have all of the different pieces together, my next step was to actually calibrate this multiscale model. And so in order to do this, what we first needed to do was to actually map these parameters. So as I mentioned before, in the signaling network model, each of these values can take a value between 0 and 1. Each of these species can take a value between 0 and 1. And so what we needed to do was to understand how these 0 and 1 values actually map to the stretch calculated by the compartmental model, as well as for the hormonal inputs, actual hormone concentrations. So these are hormone concentrations that can actually be measured in the circulating blood during with experiments. And so we did this by defining linear transfer functions for each of the 4 inputs in order to map between the network inputs to these real world measurements. And then we optimize these parameters in order to match experimental heart growth from volume overload, as well as these 3 hormone infusion experiments, so angiotensin, progesterone, and estrogen. And so as you can imagine here, this volume overload calibration will allow us to match the stretch input, and then each of these hormonal infusion experiments can allow us to map the hormonal inputs. And so this is what the fits actually look like. So by matching or optimizing the stretch parameter, I'm showing here that we can match the experimental volume overload growth. So in the symbols here, I'm showing experimental data from volume overload in rats from these different studies here. And in the line, I'm showing how we can tune the parameters or calibrate the parameters in order to match this response. And we can do the same thing for the angiotensin 2 input, as well as progesterone and estrogen. So estrogen, we actually see a little bit of a decrease, but it's a very non-significant decrease. And I should also mention here is that we are also careful to make sure that the loading was correct. So here I'm showing changes in cardiac output for these four conditions. So if it's volume overload, you get that increase in stroke volume. So the heart is actually pumping more blood into the body. So you get an increase in the cardiac output, which we're matching with the experiments. And then with angiotensin 2, you actually get a decrease in the cardiac output. So you can see that we can adjust the circulation parameters to actually match that data. And so again, what this allows us to do is to map the network model inputs that go from 0 to 1 to actual concentrations of the hormones. So I'm showing estrogen here, progesterone here, and angiotensin 2 here. And then for the stretch input, it allows us to map the network model input for stretch to the stretch calculated by the compartmental model. So this is an input into the network model, but the stretch is actually calculated by the multiscale framework. And so basically this is showing us what's happening to the elastic stretch. But as you can see here, the important part here is that in volume overload, you get the acute increase in stretch, which then gradually goes down as growth occurs. Whereas in the other conditions, in the hormonal infusion studies, we don't see that much of a significant increase in stretch. And this makes sense because we're not actually inducing any kind of overload, mechanical overload in these studies. And so basically what this allows us to do is to say, okay, if we induce this much amount of increase in stretch, we get this response in the heart growth. And then if we infuse this particular level of angiotensin, progesterone, and estrogen, this is actually what happens to the heart growth, or this is how much heart growth we can expect. And so our next question was to see if our model could actually predict growth during pregnancy. And so in order to test this, what I did was to actually implement pregnancy level changes in the estrogen, progesterone, and then angiotensin. So you can see that there's experimental data to show that angiotensin actually goes up slightly towards the end of pregnancy. And so these are the network inputs or the hormonal inputs, but we also accounted for that drop in resistance, systemic resistance, as well as the increase in the blood volume. So I'm not showing this explicitly here, but this is actually what we did. We implemented those changes in that circulation model that I showed previously. And so once we introduced these changes, we were happy to see that the model actually did pretty well in predicting organ level growth during pregnancy. So this is showing the change in left ventricular mass. So the star symbols here are showing experimental data from pregnant rats. So they observed about a 20% increase in LV mass during pregnancy. And then the line here is showing our model predictions. So you can see that we're actually doing pretty well in capturing at least the amount of growth of the at the organ level during pregnancy. And then on the right-hand side here, I'm just showing that we're also able to match that increase in cardiac output. So this gives us some confidence that we are getting the loading correctly, as well as the growth. And so the main advantage of this type of model is that now we can actually go back into the network model to really understand these interactions between the hormones and the mechanics. So in this particular animation here, I'm showing how the different species actually changed during the pregnancy simulation. So the pink is indicating that there's a rise in activity, and blue is actually indicating that there is a drop in the activity. So you can see that with progressing pregnancy, you can see that cell area actually increases in pink because it's actually increasing in size. And so we can actually plot each of these species throughout time. So these are the three inputs that we did or that we implemented. And then this is actually what happened to the stretch. So again, stretch is something that's calculated by the multiscale framework. And the really surprising thing here for us is that the stretch input actually did not change that much during pregnancy. So you can see here that this line is pretty flat. We weren't actually expecting this because pregnancy is associated with this volume overloaded state. So we thought that that volume overload would actually lead to increased stretch during pregnancy. And so we asked the question of, well, why isn't it changing? And the reason why it's not changing is because in this framework, stretch is dependent on how fast the heart is actually getting bigger in size. So how the unloaded geometry is actually increasing compared to the rate of increased loading. So how much that stress blood volume or the blood volume is expanding in the heart. And so when these actually track together, the elastic stretch actually doesn't change that much. And so this was indicating to us that this was actually occurring during pregnancy. And so this was occurring in this simulation because of this increase in rise in progesterone that occurs during pregnancy. So again, progesterone induces heart growth. And so this increase in progesterone was actually enough to track with the increased loading that occurs with the blood volume expansion. And so to us, this suggested an important role for progesterone in pregnancy-induced growth. And so for me, this is a really interesting framework because it actually allows us to ask questions about, is there a particular balance between hormones and mechanics that should be maintained for a healthy pregnancy? And could this balance somehow be off for particular conditions, including heart failure during pregnancy? And then the other aspect that I wanted to talk about here is that there are particular conditions that haven't been tested that we actually want to test using this framework. So in particular, the combination of volume overload and progesterone, so increased volume overload and increased progesterone hasn't yet been tested experimentally. But this interaction is actually very important for pregnancy because you're seeing that increase in progesterone as well as that increase in volume overload. And so I'm really interested in actually experimentally testing this condition and seeing how that matches with our with the current version of our model here to fully validate the model. And then also applying this framework in the future to actually understand heart failure associated with pregnancy. So a particular condition called peripartum cardiomyopathy is heart failure that can occur towards the end of pregnancy or after the moms give birth. It's a very alarming and sad condition because a brand new mother will actually have to go on a left ventricular assist device in order to try to reverse that process. So I think this will be a really great framework in trying to understand how does the mechanical loading and the unloading that especially occurs after pregnancy or after delivery, what does that look like in a normal pregnancy versus a peripartum cardiomyopathy condition? So with that, I'd like to acknowledge my lab at the University of Virginia, led by Jeff Holmes, my postdoctoral advisor. And then I also want to acknowledge Anna Estrada, who was a recent graduate of our lab, who's at Yale now doing a postdoc, as well as our collaborator, Jeff Sosserman, at the University of Virginia. And second, I'd like to thank you all for listening. I'd be happy to take any questions, and please feel free to contact me by email. It's listed here or on Twitter, especially if you have some ideas for applications of this framework, or if you're also interested in this particular topic. So with that, I'm happy to take any questions.
All right. Thank you so much, Kiyoko. And so if there are any questions, please go ahead and type them in. You can type them into the chat window or the Q&A window. If there aren't any yet, I will start myself and ask Kiyoko, how difficult would this be to personalize? So you were saying that this doesn't have a huge finite element, computationally expensive component. So is it possible to turn this into something that you could use personalized to a woman in a pregnancy situation and make a diagnosis based on it?
Yeah, definitely. And that's exactly the reason why our lab is really interested in using this framework, is because it is really actually well suited for personalizing. So it's really easy to actually change the size of the ventricles. So we can make measurements in patients. We can take measurements of their heart and actually tune the model, like the compartment, to actually match that. But the other important thing too is that we can match personalized hemodynamics. So we can take pressure measurements, for example, using a catheter, or even just blood pressure measurements in different patients, and actually tune the resistance and the capacitors to actually match that condition. And so I think that this framework is actually very well suited for personalizing. And that's something I'm really interested in doing too, because right now with pregnancy, and especially in the first pregnancy, all women are kind of treated in the same way. But I think, if we can get some information about a woman's biology, but then also her anatomy, we can actually take into account both of those effects using the Multiscale framework. So I think it'll be really great for personalized medicine, especially for pregnancy. I think that would be really exciting.
And is there something you think could be done interventionally? So if you did realize based on your modeling that there was something going wrong with the pregnancy, is there something, do we have the ability to intervene?
So not yet, from my understanding. So I have a couple of ideas. So I think one thing that would be very valuable is if we can actually make predictions of which women are going to experience a particular condition. So people often describe pregnancy as a cardiovascular stress to the mother, and it can actually uncover unknown cardiovascular diseases that a woman didn't actually know that she had. And so actually using this framework and kind of trying to simulate each woman, for example, and to try to understand, is this woman going to actually experience heart failure? I think that would be very valuable. But then on the intervention side, I think that's also possible with this framework. Probably not in humans at first, but at least in animal models. So for example, using the network model, we can simulate different genetic knockouts. So we can knock out a particular node that we think has a very important role in the signaling. And then we can actually test that in a mouse, in a genetically modified mice. And then, we can also use that to build up the network model too. So I think for interventions, that'll be kind of the first step. And then trying to, look at drugs that can actually prevent some of those conditions from happening.
So use it as a personalized research tool instead of as as a personalized diagnostic tool in the first instance.
Mm-hmm.
Okay, we've got a question coming in. Are there any new pathways or variables to this model that you would like to add in the future, and why?
Yes, so that's an excellent question, something I've been thinking a lot about, and I'd love to have people's ideas on how to do it. So I would actually really like to expand the network model. I alluded to a larger scale network model that already exists for cardiomyocyte or heart muscle cell hypertrophy. And so this model is like much more expansive. It incorporates 14 different pathways that are actually known to induce changes in heart mass or growth. The problem right now is that there's just not that much data. So there's some data on estrogen. People have been interested in that because of postmenopausal heart failure. But progesterone, I could only find 2 or 3 papers that actually tested cardiomyocyte reactions to progesterone. And so that's something that I'm really interested in focusing on for my research. I think, some of these stem cell platforms that are coming up would be interesting, like experimental platforms to look at. But if you're interested in doing this experiment, I would actually really love to talk to you because that's really what's missing right now, is that we just don't know how those hormones should wire into that larger scale network model.
And so it fits in with our general problem in pregnancy research of just not having enough data.
Yeah, but I think it's really important. One of the big things that's coming up now is that we don't know how pregnant women respond to a vaccine because we can't do these studies on women. And so I think it's a really important problem and a really important platform, because then, what if we can actually simulate some of these drug interactions that we can't actually ethically test? And so I think it's important. So if you feel the same, please reach out to me.
Excellent. I think I'm going to cut off the questions now. Thank the questioners and thank Kiyoko again for her presentation, which was absolutely excellent. But I don't want to miss the opportunity to get the second half of our presentation here. And so our second speaker today will be Alys Clark from the University of Auckland. And I first ran across her in a modeling workshop in Manchester over in the UK, where a whole bunch of us were talking about how to model placentas. And so she has been doing, probably, as far as I can tell, some of the best work in the world on this topic of modeling placentas, working closely with experimental collaborators to build the data necessary to feed into the models and to develop this sort of in silico approach to go forward with the idea of being able to simulate some of these things that are really difficult ethically to test. And so with that, I will happily turn it over to Alys. Go ahead and put your slides up. And again, if there are questions, please do enter them in the chat window, and we will definitely get to those.
Thank you, Michelle. So as you just mentioned, I'm interested in modeling and measuring the placenta and the uterus. Today, I'm going to talk about the uterus predominantly, but we're doing a whole line of complementary research in placental function. So before I start, I just have to acknowledge that I could not do this alone. I am a computational modeler by trade, and the work that I'm presenting here is done as part of a joint research group between the Auckland Bioengineering Institute and the Faculty of Medical and Health Science, led by Joe James. And a lot of the work presented here was done with and by a postdoc, Hannah Allerkamp, who is based between the two faculties. So the main motivation for the work that we're doing relates to fetal growth restriction. So fetal growth restriction is the situation where fetal growth it does not reach its optimal potential. So basically, the fetus starts to grow, and that growth slows down in the uterus, and the baby is pathologically small. There's many problems with diagnosing and treating fetal growth restriction. We don't have a great understanding of why it occurs, which I will touch upon in the next slide. And because we don't really know why it happens, it's very difficult to predict which pregnancies will develop growth restriction. So For example, we're doing ultrasound scanning typically at 20 weeks of pregnancy, where a baby might be tracking normally along its growth curve, but there may still be a pathology present. And then we don't necessarily do any ultrasound later on, and the baby then drops off that growth curve. So because we're not great at predicting which pregnancies will develop growth restriction, we actually fail to detect more than half of the babies that have growth restriction prior to delivery. And it's very difficult to offer treatments when we don't really understand why it happens, and we're not very good at predicting or detecting it. So our work is all about how we might be able to understand the condition better and ultimately do a better job of detecting problems. So fetal growth restriction often is a case where the fetus just doesn't get the nutrients it needs to develop properly, and that is often termed placental insufficiency. And placental insufficiency can happen because the placenta, which is the fetal organ that functions as an exchange organ to get oxygen from mother's blood, doesn't develop as it should. It can happen because there are issues with the uterus, which is what I will focus on today, and that leads to reduced blood flow from mum to the placenta. And finally, it can happen because the interface between the uterus and the placenta is not sufficient for efficient exchange. So within our group, we're aiming to provide new ways to improve pregnancy outcomes by understanding placental and uterine physiology and pathophysiology, so how the maternal and fetal circulations interact with each other, and trying to understand the anatomy of that system, which is very difficult to observe in ongoing pregnancies, and try to work out which aspects of that system have the most functional importance. And ultimately, we want to use this knowledge to improve interpretation of clinical imaging. So we do this by creating virtual representations across multiple scales via the concept of the Physiome project, a virtual representation of those organs. We use these virtual representations to relate measurements taken at different scientific disciplines, which I'll talk about today. And ultimately, we're hoping that we can use these models of the uterus and the placenta to predict the impact of therapies via virtual clinical trials. So just a little crash course on the uterus and how it changes in pregnancy. So the uterus is full of blood vessels. It's got a complex network of blood vessels within it. Starts at the uterine artery, which branches out to arcuate radial arteries, and finally into these tightly coiling spiral arteries, which are very, very high resistance. And so when somebody is not pregnant, they don't get a lot of blood flow. But when pregnancy occurs, the placenta attaches to the uterus, and these spiral arteries, which were tight coiling arteries, are remodeled into wide funnel arteries, and the whole of the rest of the uterine vasculature also remodels. There's an increase in the size of those vessels to account for a 15-fold increase in blood flow to the uterus in pregnancy. So that vascular remodeling can be termed trophoblast dependent and trophoblast independent. I'll just briefly touch on what that means. So I mentioned before there were these really tightly coiled spiral arteries in the uterus. And in the early stages of pregnancy, trophoblast, which are fetal placental cells, invade into the uterus and into these arteries. They actively destroy the smooth muscle that lines the wall of these arteries, and they go from these coiled, highly muscular arteries to more passive funnel-shaped arteries. So that's right at the interface between the uterus and the placenta. So the rest of the blood vessels in the uterus also increase in size dramatically by up to 2-fold, but they don't get actively invaded by trophoblast from the placenta. So the things that kind of drive that remodeling are hormones, which are hormones of pregnancy, estrogen, progesterone, and so on. And also physical forces. So the fact that there's an increase in blood flow and potentially pressure into the uterus influences the active contraction via nitric oxide pathways of the blood vessels within the uterus. So trophoblast-dependent remodeling, that's the active spiral artery remodeling by placental cells. It's been the focus of a lot of attention for many years because it's very dramatic. And there's very clear evidence that the fact that these arteries remodel into these wide funnels does is it prevents this a really high velocity flow of blood, which could damage the placenta that lies below it if the arteries were very small and muscular. And it allows for really good perfusion of the placenta and really effective exchange. But when we're actually looking at clinical investigations of the uterus, the measurement of uterine function is made at the main uterine artery, which is way up beyond those trophoblast-independent remodeled arteries. It's the largest artery in the uterus. And Doppler is normally performed in that artery. In normal uterine artery Doppler, in the first trimester, we would see an increase in blood flow followed by a decrease and then a second increase called a notch, which reflects a high resistance uteroplacental vasculature. And as pregnancy progresses, we start to lose that notch, showing that the resistance of the uteroplacental vasculature has decreased. And so the presence of that notch remains in some pathological pregnancies, and it's been correlated with inadequate spiral artery, so trophoblast-dependent remodeling, but it's correlated, there's no causative link between the two. So the interpretation of Doppler ultrasound, it actually depends on an old mathematical model, which describes the transport of blood from the uterine artery, the main uterine artery, which is a relatively long artery, to the uterus and then to the placental bed. And that the whole of the uterine blood vessels and the placental bed in that model are represented by a single resistance and a single capacitor, a single load impedance, assumed to be dominated by the spiral arteries, which are these dramatically remodeled arteries down here. But that model neglects all of this information on the anatomy that lies upstream, and it's this major artery right here in which Doppler is being measured. So we set out to include that anatomy explicitly in a model of uterine function. And what we were able to show in that model is that these arteries really matter. Particularly, there's connections between arteries and veins, anastomoses, that lie here in the system. So when there's no anastomoses between arteries and veins, so you just have a direct connection from the uterine artery to the placenta, if you're increasing spiral artery resistance, what happens is you get a decrease in the flow velocity, and eventually you start to see notching in the predicted Doppler waveform. But as soon as you put these connections between arteries and veins up there, which are basically a short circuit, we see that you can increase this resistance of this spiral artery dramatically and not really see a difference in what you're seeing in Doppler. So we're predicting with our model that you can get a normal arterial waveform with quite an inadequately remodeled spiral artery that's feeding the placenta directly. But the larger uterine arteries, so those radial arteries upstream, actually can result in abnormal Doppler signals. So if we have a normal-sized radial artery down here, we have no notch in our waveform in late gestation. But if we reduce the size of that radial artery, which sits up here, so this is the placenta and this is the uterine artery where the Doppler is being taken, we start to see notching in our waveforms, which is a classical sign of an abnormal Doppler, ultrasound Doppler. And critically, when we start to include the whole anatomy with the anastomoses between arteries and veins and all of the different levels of arteries, we're able to actually explain post-delivery function. So if we only had spiral arteries dominating our model, so those are the trophoblast-dependent remodeling, those actually disappear when at delivery they clamp down and there's no blood flow going to the placenta. But we still see a persistence of pregnancy-like ultrasound after pregnancy. And that can actually be explained by these larger arteries upstream of the placenta. So around about the same time as we were doing this modeling, other groups were looking into what is happening to those spiral arteries and the arteries of the uterus in early pregnancy. And they found that there was quite dramatic changes in the spiral arteries. So there's channels, there's plugs that exist in those spiral arteries in early pregnancy, which channels form through them from 6 to 8 weeks. So there should, in theory, be significant flow through them. But when you look at contrast-enhanced ultrasound, there simply isn't at that stage of pregnancy at 6 to 8 weeks. There's no real increase in flow to the placenta. And that happens later at about 13 weeks, which just happens to correlate with when we start to see changes in the radial arteries, those larger arteries upstream of the spiral arteries. So this modeling and experimental work led us to think that perhaps there's more to those larger arteries that don't change as dramatically in pregnancy. And maybe that's what we're actually seeing in Doppler when we see abnormal Doppler signals. So we set out to try to answer, why do those larger radial arteries matter, and how does flow through them actually affect blood flow to the placenta? And we've been working under a working hypothesis that those spiral arteries, those dramatically remodeled arteries down here, they change the speed of blood flow to the placenta. So they prevent high velocity jets of blood flow to the placenta. But these larger arteries, specifically these ones around about here, the radial arteries, potentially are the rate limiting step for the total amount of blood that can reach the placenta. So they're what are determining whether that 15-fold increase in blood flow to the uterus can actually happen in a normal pregnancy. So kind of thinking of it as feeding your garden bed. You have like a sprinkler on the end of your hose, which is representing your spiral arteries. And ideally, they'd be nicely dilated to allow a sprinkling over your little seedlings that you're growing rather than a jet, which would remain if they were very small. But the rest of your hose is actually determining how much blood and at what pressure, or how much water in the case of watering your garden, or what pressure you can actually feed your seeds. So we've proposed some new solutions to this problem. So the uterus is really very inaccessible in pregnancy or otherwise. But we need to know more about the anatomy and how that changes in pregnancy. So we're proposing to collect new anatomical data in both human and animal pregnancies so we can compare the two. And using in silico modeling, so computational modeling, and functional data on how those arteries, those larger arteries actually remodel, we can start to understand how that system evolves in early pregnancy. So the questions we're trying to answer is really what drives the anatomical changes in the uterus at that stage of pregnancy, and which arteries really matter. So the first thing we needed to do was really to understand more about what happens in the vasculature of the uterus in the early stages of pregnancy. And like I said, it's really hard to access that in humans. And we were very lucky to be able to visit University of Cambridge to actually digitize historic slides of uteruses from 6 to 20 weeks of pregnancy. And so what we were able to image digitally was slides which show the uterus, and in green here you have arteries circled through the uterus and the placental attachment to the uterus. And it's a story for another day, but we've also recently been able to take slices that are taken in sections, one after the other, and very misaligned and very old. These were collected in the 1950s and 60s, and align them to create 3D models of the structure of the uterus from that historic imaging using machine learning. So using that data, we were able to start thinking of the arteries that lie upstream of the spiral arteries. So the radial arteries and the arcuate arteries up here. We were able to show that there's an increase in twofold between 6 and 20 weeks of pregnancy in the size of both the arcuate and the radial arteries. And the spiral arteries, they change dramatically in size. They grow dramatically up to the second trimester. And what happens there is they become bigger than the radial arteries. So that's providing pretty strong evidence that these radial arteries up here are now the rate limiting factor towards the second trimester. They're really going to tell us how much flow is getting to the placenta. Because animal models are easier to access than human models, we're also trying to look at the same 3D structures in rat models via micro-CT. So we can separate out in rat models the uterine artery connecting to the arcuate and radial arteries and through to the placenta. So now we have new information on the anatomy in both rat and human pregnancies. We can update network models which reflect the differences in structures between those two species and with real meaningful anatomic data. So each level of artery in the system can be represented by an individual resistor and a capacitor, which allows for a fairly simple prediction of Doppler waveforms in the two animals, but also the distribution and stress in each of those types of arteries, the distribution of flow and stress. So we're able to artificially perturb the diameter of those radial arteries that we think are important and look at the impact of those radial arteries on both flow through them and the shear stress on their walls. And then we're able to compare human and rat arteries in a similar context. And so although there's a lot more blood flow through an individual human artery, the arteries are quite a lot bigger. We're predicting there's actually quite similar shear stress between the two. And shear stress drives vascular dilation via nitric oxide. So that's quite an important way to compare the two species when you're trying to interpret data that's collected in rats and infer information about humans. So then we can zoom down a step and start to think about isolated arteries. So we're conducting pressure myography experiments in rodent models. We're trying to understand what is the influence of pregnancy on the response of an individual artery to flow and pressure within it. So we're just taking a conceptual model here that the mechanics of an individual artery depends on its passive vascular structure. So if there was no smooth muscle activated in the arterial wall, it has a passive mechanical function. The pressure in the arteries, the blood pressure, the transmural pressure, also via the myogenic response, impacts the arterial mechanics. There's an active response to wall shear due to the changes in flow through the artery, which are significant in pregnancy. And then there's hormonal influences. So if we do the same myography experiments in non-pregnant and pregnant arteries, we have one set of arteries that are not under the influence of pregnancy hormones and another set that are. And so far, we have been able to complete pressure myography looking at the passive vessel mechanics, the response to intravascular pressure in both pregnant and non-pregnant rats. And we're on our way to understanding a little bit more about the shear stress response, with the future being trying to look at dose responses to pregnancy reflective hormones and try to understand how different hormones would influence the mechanics of an isolated vessel. So in a simple conceptual way, you can think of the tension in the vessel wall as having some passive component and then some active component, which is the sum of a response to pressure, a response to wall shear, and then potentially hormonal influences. And we can collect data in pregnant and non-pregnant arteries and look at each of these individual responses together. So the passive vessel mechanics in a pregnant rat artery versus a non-pregnant rat radial artery are actually quite different. So we're by fitting a model to understand passive vascular mechanics in these arteries, we're able to show that the compliance is higher in pregnant arteries and the passive lumen diameter is actually larger. So we have bigger arteries which dilate more and due to the same pressure. So we're seeing that potentially those arteries are allowing more flow just passively into the uterus. If we look at the response to intravascular pressure with smooth muscle function, although we see a shift of our curves, we're actually seeing that the pregnant arteries exhibit a comparable smooth muscle response to non-pregnant arteries in response to a change of pressure. And theoretically, we can then start to say, well, what if, because mum's cardiac output is increasing, there's more blood flow going to the uterus, how does that shift our curve? So we have the passive response and active response in terms of pressure only. And then the fact that you're getting increased flow through the system kind of starts to bring the two lines together, the red line in this graph. So where are we? We're heading with all of this. I've kind of talked separately about the anatomy and the function of isolated arteries. So we're trying to really do a good job of quantifying that anatomy because it really is unknown, certainly in human pregnancy in early gestation. And then we have these individual models of isolated vessels that can fit within that system. And so the next step is to take those vascular mechanics models and embed them within models of system function. So these kind of compliance and resistant networks of the system, but also in more anatomical fluid dynamics models of the system. And so the ultimate goal here is to try to understand, first of all, how that anatomy changes in a normal pregnancy and how that relates to how an individual vessel is then responding, which relates back to what we see next in the anatomy. So what is really driving those changes in vascular structure and function in early pregnancy? What is going wrong potentially in growth-restricted pregnancies? And where do we need to look? Potentially in Doppler, do we need to image the radial arteries or a different part of the system, or do we need to image in a different way to see those problems sooner in pregnancy for diagnosis? And so I just want to end by thanking, first of all, the team at University of Auckland, particularly Joe and Hannah, who have been very heavily involved in this work. Jonathan, our master's student, who is doing the 3D reconstruction of the uterus, and Tet, who has been really helpful at writing code throughout all of this project. And Rojan, who did a lot of the preliminary work for us to start to even think about those larger arteries, and also to the rest of our group, as well as funding from the Marsden Fund, Sally Collins and Gordon Stevenson, who have really been helping with translating this to ultrasound, Terry and Jess, who've done a lot of work in animal models, and Professor Graham Burden, who gave us access to that really great resource in Cambridge. Thank you.
Beautiful work. Thank you so much for that presentation. I will ask people to ask questions again by typing them in. And in the meantime, I will take advantage of my opportunity to ask a question myself. What limitations do you have with getting the information from the ultrasound Doppler? Is there something that you could get from MRI, or is the limitation of resolution of what you can actually see with imaging modalities in living patients kind of is that limiting what you can do here?
Yeah, so that's a great question. So ultrasound, typically you would be just looking in Doppler in the major arteries, but there's been a whole explosion of ultrasound technologies over the last 5 years, probably. And with power Doppler ultrasound and potentially high frequency probes, you can actually see quite a lot of blood vessel structure within that uterine wall. The problem is that while we have a qualitative understanding of the anatomy there, we haven't really had quantitative understanding of that anatomy. So it's very hard to know what you're looking at when you're looking at those ultrasound images where you're seeing that vascularization through the vessel walls. So this will contribute back to that kind of understanding via Sally and Gordon's work to try to test whether what is being seen in ultrasound is actually reflective of that anatomy that we're seeing in those historic samples. In an MRI, there are quite a few studies kind of coming out looking at perfusion of the myometrium, so a averaged kind of flow. The only way you can really see the anatomy is via contrast agent administration, which is not really going to happen in human pregnancies. So the resolution is just not there really in MRI, but it's coming. And hopefully having this new understanding of the anatomy will really help to drive better imaging.
Great. Well, we have a question coming in. Are there interactions between the uterine arteries and uterine muscle cells during pregnancy? And could these interactions affect the mechanical functioning or remodeling?
Another good question. Probably, yes. I'd say that there are. So yeah, we're definitely looking so far at these isolated arteries and then kind of embedding these arteries into a network of blood vessels, but without really considering the tissue that they're tethered to. And I know for sure that there will be changes in that tissue structure, and a lot of stretch in the uterus in pregnancy. And so, yeah, as a system, that will be really important, and that would be work which would be really cool to do in the future.
Do you think that we have the computational power that we need to solve these problems, but we don't have the imaging information, or do we still not have the ability to model things at the resolution or the multiple scale that would be necessary to understand this better?
Yeah, so I think there's some really good examples outside of pregnancy of this kind of multiscale modeling. Although, I mean, there's still some gaps, and probably less in the computing power and more in the logistics of how you link all the different information together. But for sure we're missing information in pregnancy. We're collecting lower resolution and less data in pregnancy, and that is an issue. And it's been really great to work with Joe and the team in obstetrics and gynecology and with those isolated vessel models to actually just really separate out all of those factors. And hopefully there's more good things to come.
We've got another question that just came in. Is any of the animal research being done in an ovine model rather than in mice or rats?
Not in our group. So, I mean, the human placenta and uterine vasculature is really quite unique. The extent of remodeling and the extent of trophoblast-derived remodeling is unique. And also the placental structure is quite different between different animals. So in rat models and mouse models, you have more of a role of angiogenesis than you would in the human models, which is much more about an invasion and remodeling process of those existing arteries. And sheep placentas are very, very different to mice and human and rat placentas. And so I think but a lot of treatments are developed in ovine models. So I think it is really important to actually understand those differences because that's kind of how you'll get the treatments translated to the human.
Yeah. Excellent. Well, if I can just take a minute to sum up, then I think both of these presentations have been excellent in that they've given us a really good understanding of why computational modeling is so important in this field. So there's limitations in the data that we have. So there's more experiments that need to be done. But then we run into the fact that there aren't good animal models, that there are ethical limitations in studying these things in humans. And so a really good in silico model of pregnancy is actually probably going to be the only way that we can test some of these things, and that we can develop better understanding and not only of understanding what's going wrong, but being able to intervene. And I think that's been the holy grail of pregnancy research for some time is if you understand what's going wrong, can you do anything about it? And we're still stuck in the if we understand what's going on part. But hopefully with time, we'll get these models going in such a way that we can start doing interventions. So with that, I would like to thank Kiyoko and Alys for their excellent, excellent presentations. Thank you to the audience for listening. And thank you very much for the questions. And I hope everybody is really interested and excited in all of the future of pregnancy research and bioengineering. And have a happy International Women in Science and Engineering Day.
Thank you. Thank you very much, Michelle, Alys,
Thank you,
and
Michelle.
Kiyoko. It was an amazing webinar, and I want to thank you all for watching and going a little bit above the hour. And I think it was a great discussion, great presentations. We'll look forward to the next one. We're going to have it in about a month or so, to be announced on Twitter. And until then, I hope you can all check out our YouTube channel, our Twitter channel, and keep posting all the great developments at JMBBM and submit your papers. And thank you all for the reviewers that do the hard work in making sure peer review is effective. Thank you very much, everyone, and see you soon.